In a studio on the south side of Chicago, an artist stands over a flat panel, pouring resin onto wet paint. Bruce Riley works in layers, dripping paint, manipulating it while still wet, then flooding the surface with clear resin before the colours can dry. Then he does it again, and again, layer after layer, building depth that catches light from multiple angles, creating shadows deep within the artwork. His paintings grow without preconception, in what he calls the biomimetic state, allowing the work to determine itself through process, through flow, through the interaction of materials that he can guide but never fully control. The results look like something pulled from a microscope. Organic forms blooming across the canvas, psychedelic mandalas that resemble cellular structures, patterns that seem simultaneously alien and familiar, like peering into a petri dish where life has just discovered colour. I'm addicted to the psychological risk-taking that is part of unknowing, Riley says. When I work too much from a place of intent, the life leaves a piece. He's describing his art, but he could just as easily be describing evolution, because if you zoom in far enough, if you look closely at the actual petri dishes in laboratories around the world, you'll see Riley's paintings coming to life. Bacteria spreading across agar in blooming fractals, cells dividing, aggregating, forming structures that no single organism planned. Patterns emerging from simple rules applied by billions of individuals. The resemblance isn't coincidental. Riley's process, layering, flow, emergence, letting complexity arise from simplicity, mirrors the fundamental process that invented you. That invented every multicellular organism on Earth. Right now, beneath your feet, inside your body, in every drop of water and handful of soil, invisible communities are negotiating the same bargain that made complex life possible. Individual cells are deciding stay alone or come together, compete or cooperate, remain independent or sacrifice autonomy for something greater. This is the most fundamental question in biology, and the answer is written in every cell you're made of. 600 million years ago, cells faced a choice: stay alone or come together. And those that chose cooperation invented something the universe had never seen: multicellular life. You're watching the descendants of that choice right now. Every breath you take, every thought crossing your mind, every heartbeat echoing in your chest exists because microscopic cells learned to trust each other, to communicate, to sacrifice individual freedom for collective survival. But here's what haunts biologists about this transition. In every act of cooperation, there's temptation. Cheetah cells that take the benefits without paying the cost. Freeloaders that could destroy everything. So how did cells overcome their selfish nature? What invisible force convinced them to build bodies instead of staying free? This is Omni. If you're fascinated by the hidden mechanisms that built complex life from simple beginnings, subscribe and join us as we explore the invisible worlds that made you possible. Tonight, we're tracing the origin story written in microscopic ink. We'll follow the moment when single cells stopped dividing apart and started holding together. When they evolved the molecular glue of cooperation, the language of chemical signals, and the ruthless mechanisms that punish cheaters. We'll watch bacteria form biofilms that behave like primitive organs. We'll meet organisms that flicker between solitary and social states. And we'll discover that the boundary between me and we is more fragile than you ever imagined. Because understanding how cells learn to cooperate isn't just biology. It's the story of every complex thing that's ever existed. From a slime mold's tower to a human brain. From a beehive to a civilization. From Bruce Riley's paintings to the body watching them. The rules are older than animals, older than plants, older than anything with a face or a name. They were written by cells, in the invisible dark, learning to become something more. The story begins in darkness. Not the darkness of night, but the ancient, absolute darkness of an ocean 4 billion years old. No eyes exist yet to see, no minds to wonder. Just chemistry, endlessly cycling through the water. And yet, even here, something remarkable was happening. A single bacterial cell, invisible to any imagined observer, was releasing molecules into the water around it. These weren't waste products or accidents of metabolism. They were messages, chemical sentences written in the universal language of molecular shapes, drifting outward from the cell like ripples on a pond. But here's the strange part. The cell was talking to itself, or more precisely, it was counting, measuring how many of its own kind surrounded it in the darkness. When those signal molecules accumulated to a certain threshold, when enough cells were present, something changed, genes switched on, behaviours transformed. Individual bacteria, acting alone, suddenly became something else entirely, a coordinated collective. This process, which scientists would eventually call quorum sensing, allowed bacteria to do something that seemed impossible for creatures without brains or nervous systems. They could assess their population density. They could wait for the right moment. They could act in unison. Think about what that means. A bacterium measuring a millionth of a metre across with no neurons, no awareness, no sense of self, had evolved the ability to know when it was alone and when it was among family. And it changed its entire life strategy based on that information. The ocean was full of these invisible conversations. Bacteria in the water column secreted autoinducers, signal molecules that accumulated as populations grew. When the concentration reached a threshold, when the quorum was reached, entire communities would synchronize their behavior. Some would begin glowing, creating bioluminescent displays that could be seen from the surface. Others would start producing toxins or digestive enzymes that no single cell could manufacture effectively alone. The chemical signals varied between species like different languages. Gram-negative bacteria produced acyl homoserine lactones that could passively drift through their thin cell walls. Gram-positive bacteria used peptide signals that had to be actively transported across their thicker peptidoglycan barriers. Each species had evolved its own dialect, its own frequency, its own way of saying, "we are many." But the most extraordinary thing wasn't the communication itself. It was what the communication enabled. When bacteria sensed a quorum, they would begin producing what scientists call public goods, molecules that benefited the entire community, exoenzymes that broke down complex nutrients, components of biofilms that created protective shelters, virulence factors that overwhelmed host defenses. These were expensive to make. They cost energy and resources. And here's the problem. Any cell that didn't contribute, any cheetah that saved its resources while enjoying the benefits, would grow faster than the cooperators. This should have destroyed cooperation before it ever began. Natural selection should have favoured the freeloaders, the cells that took without giving. And in many bacterial populations, that's exactly what happened. Cheetah mutants that didn't produce the costly signals or public goods would invade populations of honest cooperators, threatening to collapse the entire social system. So why didn't cooperation disappear? The answer lies in something subtle, something that changes everything about how we think about individual and collective. Cooperation was more likely to evolve and persist when bacteria were highly related, when all the cells came from a single founding cell. When you're surrounded by your genetic siblings, helping the group means helping copies of your own genes. The distinction between selfish and altruistic behaviour begins to blur. Picture a bacterial mat forming on the ancient seafloor. A single cell divides. Its daughters divide. Within hours, a colony of thousands blooms into existence, all carrying identical genetic instructions. When these clones sense their own autoinducers, when they detect the quorum, they're not really communicating with strangers. They're coordinating with copies of themselves, like a distributed intelligence running the same program across multiple processors. The signals themselves were more sophisticated than anyone initially imagined. Concentrations of molecules like cyclic deguanylate would oscillate in well-defined patterns across generations, creating waves of information that spread through bacterial lineages like radio transmissions. Different frequencies, different amplitudes, encoding different messages about surfaces, about nutrients, about danger. This was mass communication without mass media, broadcasting without transmitters, a form of collective computation happening at the molecular level, where individual cells became nodes in a living network. And it enabled something extraordinary. When enough bacteria gathered in one place, when the quorum was reached, they would begin building biofilms, vast architectural structures made of cells embedded in a self-produced matrix of polysaccharides, proteins, lipids, and DNA. These weren't just random aggregations. They were organised three-dimensional communities with distinct zones and functions, channels for nutrient flow, layers with different metabolic activities, protective barriers against antibiotics and immune cells. Bacteria had invented cities, complete with infrastructure, resource distribution, and cooperative defence, all without a blueprint, without a foreman, without any cell understanding the larger structure it was helping to build. The biofilms spread across every surface in the ancient ocean. They coated rocks, formed mats on sediments, created living carpets that trapped nutrients and changed the chemistry of their surroundings. And within these structures, the real innovation emerged. Bacteria weren't just cooperating with their own kind, they were coordinating with different species entirely. Some bacteria evolved the ability to produce and detect AI2, a universal signalling molecule that could facilitate interspecies communication. This was profound. It meant bacteria could assess not just their own numbers, but the entire composition of the microbial community around them. They could detect when they were in heterogeneous environments and modulate their behaviour accordingly. Competition and cooperation occurring simultaneously. Species that normally competed for resources would share information, coordinate biofilm formation, even enhance each other's survival. Some bacterial species like Burkholderia capacia and Pseudomonas aeruginosa would use quorum sensing to amplify each other's virulence, creating devastating co-infections. Others would form symbiotic partnerships, with one species creating conditions that allowed another to thrive. The ocean floor became a tapestry of these invisible alliances, bacterial mats meters thick containing billions of cells, all communicating through molecular signals, all responding to the changing concentrations of auto-inducers that informed them about their world. Some cells at the surface received more oxygen and light. Others, buried deep in the mat, lived in anoxic darkness. But they were connected by chemistry, by the diffusion of signals and nutrients, by the emergent properties of their collective behaviour. This was the first step. The first time life discovered that many could be stronger than one. But bacteria remained fundamentally simple. Each cell was essentially identical to its neighbours. There was no specialisation, no division of labour, no sense that some cells should serve fundamentally different roles than others. The biofilms were impressive, but they were more like dense crowds than organised bodies. The next step would require something more radical. Approximately 2 billion years ago, in an ocean that was slowly accumulating oxygen from photosynthetic cyanobacteria, a different kind of cell was evolving. These were eukaryotes, cells with nuclei, with complex internal architecture, with the capacity for something bacteria could never achieve. And some of these cells were facing a crisis. Food was running out. In the soil and on decaying leaves, populations of tiny amoebae called Dictyostelium hunted bacteria. They moved independently, each one a solitary predator following chemical gradients to find prey. But when bacteria became scarce, when starvation loomed, something astonishing happened. One cell would begin secreting cyclic AMP, the same molecule that bacteria used for signalling, now repurposed for a new kind of communication. Other amoebae would detect this signal and respond in two ways. They would move toward the source, and they would release more cyclic AMP themselves. The effect cascaded through the population. Waves of chemical signals rippled outward from the initial broadcaster. Amoebae aligned themselves along these waves, forming distinct streams of cells flowing toward a central aggregation point. Within hours, between 10,000 and 1 million cells would converge, piling up into a mound. But they didn't stop there. The mound would extend itself, forming a slug-shaped mass that could migrate across surfaces. And here's the remarkable part. The cells were beginning to differentiate. Some cells in the slug would become specialised for movement, others would prepare to become spores, and still others would commit to something that seems like suicide. When the slug found a suitable location, it would transform into a fruiting body, a stalk topped with a mass of spores. The cells in the stalk would die, sacrificing themselves to lift the spores away from the ground and into air currents that could carry them to new locations. This was a form of cooperation that bacteria had never achieved. Permanent differentiation, specialized cell types, some cells dedicating their entire existence to supporting others, with no possibility of reproduction, no direct genetic future. Why would natural selection favor this? Recent research revealed something unexpected. The trigger for this multicellular transformation wasn't just starvation in general, it was specifically the loss of sulfur-containing amino acids like cysteine. When cysteine levels dropped, when sulfur became limiting, it signalled to the cells that conditions were no longer suitable for growth and division. The best strategy was no longer to remain independent. It was to pool resources, to create a structure that could survive the crisis and disperse spores to potentially more favourable locations. And the slug could do things individual amoebae could never accomplish. Experiments showed that solitary amoebae couldn't cross soil barriers that slugs navigated easily. The multicellular form was better at local dispersal, better at finding new food patches. Cells shed from the migrating slug would consume bacteria in its path, clearing obstacles. But there was still a problem. When the multicellular slug formed by aggregation, when cells of different genotypes came together, the stage was set for conflict. Cheetah cells that contributed less to the stalk but positioned themselves in the spores would have a reproductive advantage. They would outcompete honest cooperators, potentially destroying the entire system. Yet in nature, something even stranger was discovered. About 1/3 of wild Dictyostelium colonies engaged in primitive farming, carrying bacteria within their fruiting bodies to seed new locations with food. This meant accepting a cost, smaller spores that couldn't disperse as far, in exchange for a long-term benefit, arriving at a new location with your food source already present. Cooperation was getting more complex, and it was beginning to resemble something we would recognise as multicellular life. But Dictyostelium only went part way. Its multicellular phase was temporary, triggered by crisis, and the cells that formed it often came from different genetic lineages. True multicellularity, the kind that built animals and plants and fungi, would require something more fundamental, a commitment from the very beginning, from a single founding cell. The real breakthrough came in a different lineage entirely. In shallow ponds and slow-moving streams, tiny green spheres were rolling through the water, each one a perfect geometric marvel. Meet Volvox, a multicellular green alga barely 2 millimeters across, containing just a few thousand cells arranged in a hollow sphere. But unlike Dictyostelium, every cell in Volvox descended from a single founding cell. No aggregation, no temporary coalitions, permanent commitment from the very beginning. This changes everything. The Volvocene algae, the family that includes Volvox, represents something extraordinary in evolutionary biology. They span the entire spectrum from unicellular ancestors to fully differentiated multicellular organisms, and they did it recently enough that we can trace the steps. The group includes Chlamydomonas, a single-celled alga that swims alone, then Gonium, a flat colony of cells, all identical, then Pandorina and Euderina, larger colonies still without specialized cell types, and finally Volvox, with its dramatic innovation, a complete division of labor between two cell types, germ cells and somatic cells, reproductive gonidia that would create the next generation, and smaller somatic cells with flagella that would never reproduce but would spend their entire existence swimming, propelling the sphere through water, supporting the reproducers. This was the first time in evolution that cells born from the same parent committed to fundamentally different fates, not because of environmental triggers or starvation stress, but because of their position and size during development. During embryonic division, some cells divided asymmetrically, producing large-small sister pairs. The large cells activated one set of genes, the small cells activated another. Size determined destiny. The large cells would never swim. They would grow, divide, and create the next generation. The small cells would never reproduce. A gene called Reg-A would activate in them, shutting down all aspects of reproductive development permanently. They became somatic cells, body cells, condemned to mortality, existing only to serve the immortal germline. Why would natural selection favour this sacrifice? The answer emerged when scientists compared Volvox to its relatives. The entire lineage showed a progressive pattern: increases in cell number, increases in body size, increases in the extracellular matrix surrounding cells, and increasing specialization. Larger size provided advantages. It made organisms harder for predators to consume. It allowed them to stay suspended in the water column more effectively. And critically, Larger size provided an advantage in competing for limiting resources like phosphorus. But there's a physical constraint. A cell can't be both optimized for reproduction and optimized for movement. Flagella require tremendous energy and space. Chloroplasts for photosynthesis require different cellular real estate than the metabolic machinery for rapid growth and division. You can specialize for one or the other, but not both equally well. Analysis of gene expression in Volvox revealed the stark dichotomy. Gonadial cells expressed growth-related genes, ancient genes focused on reproduction and resource acquisition. Somatic cells expressed a more specialized program, younger, lineage-specific genes devoted to an altruistic metabolic program. They manufactured flagella. They converted storage carbon into sugars for producing the extracellular matrix glycoproteins that enabled the sphere to expand to its massive size. The somatic cells were metabolic servants, working for the collective good at the cost of their own reproduction. But here's what haunts biologists about this arrangement. The genetic difference between unicellular Chlamydomonas and multicellular Volvox is surprisingly small. Despite 200 million years of independent evolution, their gene content is nearly identical. The transformation from solitary cell to multicellular organism with differentiated cell types didn't require vast new genetic innovation. It required rewiring, taking genes that already existed and expressing them in new patterns, at new times and new combinations. Some of the cell type-specific genes in Volvox appeared to be co-opted from a unicellular ancestor's day-night cycle. Genes that were once expressed during dark phases became specialized to somatic cells. Genes once expressed during light phases became specialized to gonadia. Temporal programs, daily rhythms, were converted into spatial programs, permanent cell types. This is how evolution builds complexity. Not from scratch, but through tinkering, repurposing, taking systems that served one function and discovering they could serve another. And once the germ-soma division appeared, once some cells committed to dying so others could reproduce, natural selection could sculpt both types independently. Somatic cells could evolve to be better at motility, better at photosynthesis, better at building the extracellular matrix, without any concern for maintaining reproductive capacity. Germ cells could focus entirely on growth and division, protected and transported by their somatic siblings. The colony became an individual, the cells became tissues, but Volvox was still primitive, just 2 cell types, no organs, no complex development beyond the initial division between germ and soma. For the next transformation, for the leap to animal complexity, evolution would need something more. It would need cells that could not only differentiate but could also communicate position, coordinate movement, and build 3-dimensional structures with sophisticated architecture. The path to animals began in a place both alien and strangely familiar. Ancient oceans, perhaps 800 million years ago, where the oxygen concentration was finally rising to levels that could support larger, more energetically expensive life. And in those waters swam creatures called chonoflagellates, single-celled organisms with a distinctive collar of finger-like microvilli surrounding a single flagellum. They looked remarkably similar to cells found in modern sponges. The chonocytes, collar cells, that line the interior of sponges and generate water currents for filter feeding are nearly identical in structure to free-living chonoflagellates. This wasn't convergent evolution. Chonoflagellates are the closest living relatives of animals. The last common ancestor of all animals was a creature very much like a modern chonoflagellate. A single cell with a collar and flagellum filter feeding on bacteria. But unlike their modern descendants, some ancient chonoflagellates discovered they could do something extraordinary. They could form colonies. Certain species of chonoflagellates can transition between single-celled and multicellular states. They divide but fail to separate, remaining attached through their collars, forming beautiful rosette-shaped colonies of cells arranged in spheres. 4 cells, 8 cells, 16, sometimes 50 cells, all beating their flagella in coordination. And these colonies could do things single cells could never accomplish. Research showed that colony geometry affected feeding efficiency. In certain configurations, cells and colonies could capture more prey per cell than solitary chonoflagellates. The water currents generated by coordinated flagella beating created flow patterns that concentrated bacteria near the collars. but colony formation also came with costs. Larger colonies produced bigger hydrodynamic signals, making them more visible to predators. Raptorial protozoans would capture colonies while ignoring single cells. But larger colonies were also too big for some predators to handle, rejected by ciliate predators that could easily consume individual cells. The selective pressures were complex, contradictory, pushing and pulling in different directions. Sometimes it paid to be alone, sometimes it paid to be together. The ability to switch between unicellular and multicellular states, facultative multicellularity, might have been the key adaptation. In 2019, researchers discovered a new chionoflagellate species, Chionuca flexa, that exhibited remarkable collective behaviour. Individual organisms would join together by touching collars, forming a concave sheet with flagella pointing inward for feeding. But in darkness, the sheet would invert into a ball-like shape with flagella pointing outward, allowing the colony to swim rapidly, shape-shifting in response to light, a multicellular organism capable of coordinated behaviour that no individual cell could achieve, swimming toward well-lit areas where food might be abundant, a navigation strategy impossible for solitary cells. When scientists examined the molecular toolkit of chionoflagellates, they found something astonishing. These unicellular organisms possessed many of the genes that animals use for cell adhesion, cell signaling, and even genes involved in regulating development. The genetic machinery for building multicellular bodies was already present in single-celled ancestors. Evolution hadn't invented these tools for multicellularity. The tools existed first, serving other functions in unicellular life. Cell adhesion proteins that would later hold tissues together originally helped cells stick to surfaces. Signaling molecules that would coordinate development originally helped cells sense their environment. The transition to multicellularity wasn't about gaining new capabilities. It was about keeping cells together after division and repurposing existing molecular machinery for new collective functions. Some chionoflagellates formed colonies through repeated cell division from a single cell. Clonal multicellularity, the same strategy that would define animal development. Every animal on Earth, from sponges to humans, develops from a single fertilized cell that divides repeatedly, with daughter cells remaining attached. This developmental mode was already established in our chionoflagellate-like ancestors. But the crucial question remained: how did temporary colonies become permanent bodies? How did facultative multicellularity become obligate? How did collections of cells that could separate become organisms that could never again exist as individuals? The answer required three fundamental evolutionary steps. First, cell-to-cell adhesion with an alignment of fitness among cells, keeping cells together and ensuring they benefited from cooperation. Second, cell-to-cell communication, cooperation and specialization, with an export of fitness to the multicellular organism level. And third, the evolution of mechanisms that prevented cells from reverting to unicellularity. These mechanisms were called ratchets, traits that provided benefits in a group context, but were detrimental to loners, ultimately preventing any return to single-celled life. Once cells became mutually dependent, once they divided labour so thoroughly that no single cell retained all necessary functions, there was no going back. Some cells might specialise in growing one vital molecule, while others grew a different essential compound, so these cells did better together than apart. The more specialised cells became, the more they needed each other, the stronger the ratchet clicked into place. And with each click, with each increment of mutual dependence, the individual cells became less like independent organisms, and more like parts of a larger whole. The colony became the individual, the cells became organs, the organism became irreversibly multicellular. Somewhere in the Precambrian oceans, between 800 and 635 million years ago, this transition completed itself. A lineage of chionoflagellate-like organisms crossed the threshold, their cells no longer separated after division, their colonies no longer broke apart. They had committed fully, permanently, irrevocably to multicellular life, and from that commitment everything followed. 635 million years ago, as the Earth thawed from the last great cryogenian glaciation, when ice sheets finally retreated and oxygen levels began to rise in the ancient seas, something unprecedented appeared in the fossil record. Soft-bodied impressions in sandstone, frond-shaped organisms, disc-like creatures, strange quilted patterns unlike anything that came before. The Ediacaran biota, Earth's first large multicellular communities. These organisms lived for nearly 40 million years before the Cambrian explosion, representing early experimentation with complex life forms. They demonstrated mobility, heterotrophy by multicellular animals, the beginnings of skeletonization, and the assembly of complex ecosystems. They were building bodies at scales previously impossible, and somewhere among them, or perhaps shortly before them, the first true animals emerged. These earliest animals were likely sponges, simple creatures without muscles, without nerves, without guts, but with something revolutionary: multiple differentiated cell types working together in coordinated fashion. They weren't just colonies of identical cells. They were organisms with specialized tissues, each cell type performing distinct functions that the others couldn't. Chonocytes with collars and flagella that generated water currents and captured bacteria. Pinacocytes that formed the outer covering and provided structural support. Archaeocytes that migrated through the interior, capable of transforming into other cell types as needed. And critically, a mesohyl, a jelly-like matrix between cell layers that served as a scaffold, a medium for cell migration, a three-dimensional architecture that single-cell layers could never achieve. This was different from everything that came before. The innovation wasn't just sticking cells together. It was creating a polarized body plan with water flowing in through pores, circulating through internal chambers, and flowing out through an opening at the top. The sponge had invented a circulatory system without a heart, digestion without a gut, coordination without a nervous system. When scientists finally sequenced the sponge genome, they discovered something remarkable. Sponges possessed the same core genetic toolkit as all other animals: genes for cell division and growth, for programmed cell death, for cell adhesion, for signaling during development, for recognition of self and non-self, for the formation of different cell types. The molecular machinery for building animal bodies was already present in the simplest animals. What sponges lacked, muscles, neurons, guts, would come later. But the fundamental innovation, the transition from colonial organism to integrated individual, had already occurred. And here's the darker parallel that this transition revealed. The same genes that regulate cooperation in multicellular organisms are the genes whose disruption causes cancer. Multicellularity and uncontrolled cell growth are two sides of the same coin. When cells in a body decide to stop cooperating, when they revert to selfish behavior and grow without regard for the organism's needs, they become tumors. Cancer is cooperation breaking down, its cells remembering their unicellular past, rejecting the constraints of multicellular life, attempting to return to a more ancient strategy of individual reproduction at any cost. Every gene that helps cells cooperate, that makes them divide when they're supposed to, die when they're supposed to, stay in their designated roles, is a potential cancer gene when it malfunctions. The transition to multicellularity required building fail-safes, checkpoints, mechanisms to detect and eliminate cheaters. And when those mechanisms fail, the body pays the price. This is the cost of complexity, the price of becoming more than the sum of your parts. But the benefits were extraordinary. Once animals evolved spatial cell differentiation, the ability to create different cell types in controlled locations, they could build bodies with unprecedented sophistication. Not just random aggregations of cells, but architectures with purpose. Layers that could fold, invaginate, create internal spaces. Cells that could specialize to the point where no individual cell could survive alone. But together they could navigate, hunt, reproduce with complexity that dwarfed anything single cells could achieve. The cell types that evolved in early animals were built by parceling out functions that originally existed in multifunctional ancestral cells. A single chonoflagellate-like ancestor might have been sensory, contractile, and digestive all at once. Evolution split these capabilities, gave them to different cells, and in doing so created the possibility of specialization. One cell type could become exquisitely good at sensing. Another could master contraction and movement. A third could focus entirely on digestion. Division of labour at the cellular level opened the door to everything that followed, and the ratchet clicked one final time. Animals committed to clonal development, every individual arising from a single fertilised cell that divides repeatedly, with all cells sharing the same genome. This wasn't aggregation like dictyostelium. This was a developmental program encoded in DNA, executed with precision every generation, building the same body plan from a single cell through an invariant series of divisions and differentiations. No going back, no reverting to single-cell life. The individual cell ceased to be the unit of selection. The multicellular organism became the individual. From that commitment emerged the Cambrian explosion. 540 million years ago, in a geological instant, animals diversified into all the major body plans we recognize today. Eyes evolved, shells and exoskeletons appeared. Predation became sophisticated. The arms race of hunter and hunted drove innovation at a pace the Earth had never seen. But none of it would have been possible without those first uncertain steps. Without bacteria learning to count each other through chemical whispers. Without biofilms discovering that structure emerges from simple rules applied by many. Without slime molds sacrificing some cells so others could disperse. Without Volvox committing to permanent differentiation. Without chonoflagellates forming rosettes that could swim in coordinated patterns. Every innovation built on the previous one, every transition required cells to give up something. Autonomy, reproduction, immortality, in exchange for something greater. Stand at the edge of an ocean and look at the water. Billions upon billions of cells, invisible to your eyes, are negotiating this same ancient bargain. Bacteria are deciding whether to cooperate or cheat. Algae are coordinating cell division. Microscopic animals are building bodies that will last hours, days, weeks, before dissolving back into single cells that will try the experiment again. And inside your own body, trillions of cells are maintaining the cooperation that makes you possible. Your neurons aren't reproducing, sacrificing their genetic future so you can think. Your red blood cells don't even have nuclei anymore, committed so thoroughly to oxygen transport that they shed their DNA entirely. Your skin cells are programmed to die and slough away, making room for the next generation. You are a colony that forgot it was a colony, a temporary coalition of cells that believes it's a permanent individual. Your sense of self, your feeling of being a unified consciousness rather than a community of microsystems is the ultimate achievement of cellular cooperation. But the cooperation is always fragile, always one mutation away from breaking down, always requiring active maintenance, constant surveillance, perpetual enforcement of the rules that keep cells working together rather than pursuing their own reproduction. The question that haunted early multicellular life still haunts us. How do you keep cheaters from destroying the collective? Cancer is proof that we haven't completely solved this problem. Our immune system patrols constantly, destroying cells that have stopped cooperating. Our bodies execute elaborate checkpoints before allowing cells to divide, and still, sometimes, a cell escapes surveillance and begins to grow without constraint. The battle between individual and collective, between selfish gene and cooperative genome, never ended. It moved from oceans to tissues, from bacterial mats to tumor microenvironments, from simple biofilms to complex bodies. But here's what makes this story remarkable. Despite the fragility, despite the constant threat of defection, cooperation won. Multicellular life didn't just survive, it dominated. Today, the most massive organisms on Earth are multicellular. The most intelligent are multicellular. The most diverse forms of life are multicellular. The strategy that began with bacteria counting each other in darkness, that continued with slime molds forming towers, that accelerated with algae dividing labour, that culminated in animals building bodies from single cells, that strategy reshaped the planet. Forests emerged, transforming the atmosphere. Animals colonised every habitat from deep ocean vents to mountain peaks. Brains evolved, generating consciousness, asking questions about their own origins, and all of it traced back to that first moment when a cell decided not to separate from its sister after division. When two became one, when individual gave way to collective, the invisible worlds that invented multicellular life are still here, still experimenting, still negotiating the balance between self and group. In every drop of pond water, in every handful of soil, In every surface covered with biofilm, the ancient drama continues. Cells are learning to cooperate, and sometimes, against all odds, cooperation becomes something more permanent, something that can build bodies, minds, societies, something that can look back across billions of years and ask, how did we become possible? The answer was written in molecular language, in the chemical dialects that bacteria first spoke in ancient darkness, in the sacrifice of soma for germ, in the ratchet mechanisms that made cooperation irreversible, in the developmental programs that build bodies from single cells. You are the answer. Every multicellular organism is the answer. A temporary victory of cooperation over conflict. A momentary alignment of individual interest toward collective survival. A body built from billions of cells that mostly remember to work together. The invisible worlds invented you, and you carry their story in every cell, in every gene, in every molecular mechanism that keeps your cells cooperating instead of competing. Remember that the next time you take a breath, or move a muscle, or think a thought. You are not an individual, you are a community, a coalition, a democracy of cells that somehow convinced themselves they were one. And the story of how that became possible, the story of cells learning to trust each other enough to build something bigger than themselves, is the most fundamental story in biology. It's the story of how chaos learned to create order, how individuals learned to become communities, how invisible worlds learned to build visible ones. It's the story of cooperation. And cooperation, for all its fragility, for all its constant threat of collapse, remains the most powerful force that evolution ever discovered.